Everyone. Did I get my mic on? Everyone can hear me? No? All right. Which one? This? How's
No, this is her PA. Shut off in that corner. Let me try it.
How's that, better? All right, great. All right, get the hard stuff out of the way early. Right? Yeah. So yeah, Kristy, I work at Penn State. I'm also state coordinator in Pennsylvania for the SARE program, Sustainable Ag and Education. And this is a project that was started by Dr. Borrelli White, and I definitely credit. We wanted to start addressing to take a soil health. There's a lot of popularity, a lot of excitement around soil health. We know it's a really important aspect of our crop production, of crops and sustainability as a whole. And so there's a lot of tests out there and so we wanted to look at this, figure out what these we can use them in our management. Also, I wanted to point out here, we had a group of great educators involved in extension and outreach who participated in this study as well. So we know that soil health has a lot of important aspects. It helps us with nutrient cycling, mineralization of nitrogen, helping our they want. And when we do have a healthy soil, we have good water and air regulation in that soil. And then also it can help with disease and pest suppression. Some other things. And so when we look at these, there are really three different legs of a stool. We have the physical portion, the biological portion, and the chemical portion. And these are studied and understood in different ways. Some of them we've had a lot of experience looking at, and some of them they're kind of new and evolving in our knowledge and our scientific approach to understanding them. For example, the biological component is very complex, so we don't know as much about that. But when we look at them, these all break up into these different management portions. So the physical portion here, we think about things like aggregate stability, soil strength, and available water. Those are influenced by the physical properties of the soil. The chemical aspects, these are things like the nutrient test, right? We've been using these for a long time to determine how much nitrogen we want to put on our corn or how much potassium or lime. And so these are standardized. These are some of the tests that we have available now that we use regularly, and those measure things like pH, nutrients, cation exchange capacity. And then, as I mentioned, the biological component. This is an important component and extremely beneficial to our soils. However, we don't know too much about it. We know it's important, but again, it's incredibly complex. But what those biological organisms do, they're the ones that are mineralizing and making the nutrients available. They're the microbial community. We have a healthy, diverse community in our soil that are doing all sorts of things, and they also help us suppress diseases. And so they're the soil when we have a healthy soil. And so there's all these different legs of all these different stools. Some of them we. But the one thing that's in common is when we look at it, organic matter is what influences all of these. It influences the whole soil health, or the entire soil, as well as each of these factors here. So organic matter will influence the microbial community. Organic matter will influence the pH. It also will improve soil stability. So that's the covering on all of these stools. But then how do we go about this, right? Because I just presented a nice big complex system, and when we do that, we usually break it into our parts, but we also know it's interacting. So how do we go about looking at that? And so different have been looking at this over time, and there's several testing services that are available commercially. Some of the more popular ones, the Cornell Soil Health Test is available out of Cornell University. This is a very robust test, and I'm going to talk about the details about this in a little bit more in a minute. The Woods End Laboratory Soil Health Tool. This integrates the Solvita test as well as the Haney test in their analyses. And then we also have the comparison to our basic standard soil test. I'm going to demonstrate the Penn State Analytical Lab soil test today, but there's also private labs that give us information that we use. And again, these are what we've been relying on for our basic nutrient management. needs. And so how do these, so what can we learn? How are these different and how can they help us? And so one thing to look at when we, are evaluating something is something very practical. It's the price of something. And in this case, we have these different tests. And these are the different soil indicators over here on the left-hand side. And so the different tests are looking at different things. We have soil active carbon, microbial activity, amino, protein, nitrogen, soil-borne diseases, the chemistry, the compaction, and aggregate stability. And so the nutrients look at those, but some of the others are starting to examine all of these different components. And this is what you're getting when you take a soil test to Cornell. They're looking at separate components. But that does come at a cost. And so Cornell will look at all of these different, they include a summary of all of these practices in their analyses. Then does most of them. They don't look at diseases. They don't look at compaction. And one thing to point out here is
the information, how do we put it together?
Single select any one of these. So you can't go to Cornell and say, I just want to look at amino proteins. You get the whole, so there's not that option to buy them individually. Whereas the standard test, you can do that. You can get a package for your soil chemistry and then maybe add organic matter on. So standard test labs don't necessarily offer all of these aspects. So the active carbon, some of these microbial, they're just not set up to look at those quite yet. But the cost of that is, I think Cornell costs about $95, sometimes $75 for that whole package. For one soil sample, Woods End runs about $50 a test. And the Penn State, the lab, that's going to vary again based on lab, but that is about $15, and if you want to do organic matter, that's a $5 add-on. So you're talking about $20 compared to about $100 for your test. And so looking again at our stools and the importance of organic matter here, and then again considering the cost of the test, you can we look at just organic matter as a study of these soil health tests? It's inexpensive. It's already routine measured. We have a good standard in place for quantifying that. Well defined at what it is. We know it's important to all these different legs of the stools, and it's related to these processes. And so this is the research question we want to address with the SARE project. And so we set out, educators identified different farms, and we did this across the state of Pennsylvania, several different locations. And they went out to different fields and sampled. So they would either sample two different systems, till versus a no-till system, for example, cover crops versus no cover crops, or they would go out and look at the top 4 inches of the soil versus the bottom 8 inches of the soil. So they just did a comparison of two different tests or soil samples. And we also did this at there's a long-term cover crop project in Penn State at one of our research farms, and so we grabbed some soils from that too and added those into our collection to analyze. And so what we did is we went to the 3 different labs. The Woods End premium test for $75, Cornell test at $100, and the PS percent organic matter as well as the Mehlich 3 nutrients. We did that for $14. We sent 30 samples total for these correlations to look at these. And I did want to point out here, we did, although the education sample at different depths and maybe different cropping systems, we're not comparing those at all. Those are just lumped in here, and the comparison is really between these 3 tests. It doesn't matter what the treatment was on those. This is just the distribution across that data set of 30 samples. So these here, you probably can't tell that these are blue. Those 0 to 8 depth. And then over here, these are the ones that were broken up between 0 and 4 and 4 and 8 inches. And so this is just to show the organic matter in all of these samples is approximately 0 to 8 inches. We saw 2.9% median across all of those. But again, you can see there's variability. Depending on the site location, this is always going to be true in our farming system. There's going to be extreme variability. And so we did see that, but the median about 2.9. As you might expect in the upper profile at 0.4, it's a little bit higher, 3.8%, for most of that organic matter is collecting. And then a little bit lower in that profile, similar to the 0 to 8 inch. And this compared very well. This is the median organic matter for 15 years. So this was collected from Penn State Lab. This is just the median. So overall, what they've been collecting for 15 years was about 2.7% organic. So just to give a little bit of a glimpse into our of what soils look like. And so just to kind of think about these tests, you're not always comparing apples to apples. And that's something that's a bit of a point of confusion sometimes, because if you take your soil sample, you might send the exact same soil sample to Woods End and to Cornell, and they might give you different results. And that's what we saw. And this is a really great example here. Woods End reports percent loss of ignition, and this is just estimate organic matter. So when you use the loss of ignition or LOI in these, that just means that's organic matter. And this is just a test that they use where they burn the soil, or they combust it, and they weigh it before and after, and so it's just that gross loss of weight. Whereas some of the other labs, this is well established, and they found out that that's not accurate, and so some of these other labs go ahead and correct that with a factor. So this is a little bit more of a raw number, where some of those others are more accurate. And so again, you're not always comparing the same things. And so when you get a sample, sometimes you'll estimate your organic matter is quite a bit higher than it would be from a Cornell test, and that's just because of the methods we used. So that's a little bit confusing if you don't know those intricacies of the different tests. And similarly, and this is what we did when we compared the Cornell test to the Penn State test. These are very similar. Those both use that correction factor. And we saw a great correlation between these. So Cornell, the correlation there is 0.96. They were about 86% similar to one another. But it was still, Cornell was showing it was about half a percentage point higher than what we were seeing from Penn State. And this just was a little bit confusing to the educators. Got these back. How do I make recommendations? What do I talk to my these numbers aren't the same. And so it just was a little bit of an area that warranted a little bit of discussion, a little bit of explanation of different tests. And just to be careful of that when you are sending samples, if you are, that you're not always comparing the same things. And so now I want to break down different tests and look at some of those components that are measured in those soil health tests. A little bit more detail to show you what those are telling us. And so one of the first tests that we do is active carbon or permeabilizable carbon. Sometimes you'll see this as POC, OXC. And so this is correlated to the total organic matter in the microbial biomass. So basically, you take your soil sample, how much carbon is in there to be potentially essentially. So it's measuring all the carbon in that. And it does this by using a weak solution of potassium permanganate, and it oxidizes that carbon out. As the tests go, this is only available through the Cornell test, and again, you can't buy that separately. That's part of the package. But when you run it, you end up getting these very the permanganate makes it purple in color, and so it's a nice method if you have a laboratory or access to it, because you can run these colorimetrically. You don't have to necessarily go to a different lab or buy extra equipment. So if you are set up for it, it's convenient that way. Also very sensitive to short-term changes in management. So if you want to know, I did this practice, just a short time ago, you can compare it to that and see a change in these. So that has an opportunity to do that. And so this is what we saw. This is the Penn State organic matter on the x-axis, and looking at that active carbon, we see similarity of about 70% of R squared there. So that's a pretty good correlation. And this is active carbon. This is measuring fuel activity. It's very hard to pin down. And so getting an R square of about 0.7 is quite good. And so we did see a good correlation between organic matter and that. And we have a couple samples out here. These were very high. These were Packer samples. They had organic matter of about 7 to 8%, not quite 8%, but they were quite high. That's why that curve is bending a little bit. Just we had a couple outliers where all of these other systems were managed more intensively agriculturally. Okay, the second test was the CO2 burst, and this is measuring microbial activity. So this is actually the respiration of the microbes. So again, relating to carbon, similar to the test I just showed, but this is how how the microbes are respiring. So that's them breaking down the carbon. And that's what this is measuring. And they have different methods for this. They measure it for 24
And we're
hours,
going to push that data whether
6-hour period in the lab. And this is, they incubate wet soil. So they take dry soil, they re-wet it, and as that soil stimulates the microbes to start breaking down the carbon, and they breathe. And then they can capture that CO2, and that's how they're, what they're monitoring there. And so this is an indicator of the microbial population and the availability of the organic matter there to decompose. It also makes somewhat mineralization of nitrogen, because as these carbons are, or these microbes are mineralizing that carbon, they're also releasing nitrogen. And nitrogen mineralization is extremely difficult to measure too. It's something all of us would like to know how to measure more. It's just very difficult because it's based on temperature, moisture, all these things. And so this is a good way to glimpse into that. So it has a potential there to help us understand that. And this is available at all several different labs. Both the Woods End and Cornell have it. It's part of their packages, and some other labs will offer this on an individual basis for about $30 a test. And here in the bottom, maybe you have seen these. This is an example of it. This is the Solvita paddle. Solvita is a company that makes these, and so this is, they re-wetted this soil, and now it's respiring, and this paddle is going to change color. It's going to change color based on how much CO2 is produced, and then you have a key, and you check that color based on that key to see how active that is. That's how that works. And here they're just measuring it. So when they looked at the microbial respiration, over on the left is the Woods End, and that's hours for that, and the 96 hours burst for Cornell. Again, a great correlation when they looked at, compared to the loss of ignition coming out of Woods End, correlation of about 0.82, Cornell organic matter about 0.79. So good correlation there, saying, and that makes sense, right? Because what's going to influence the amount of carbon, what's going to influence the amount of CO2, it's going to be the organic matter. It's directly related to that. So we did see when you measure, when you measure just the organic matter, there was a correlation in there. Long-term research study for crop management. This is done in the state. There's 7-8 different cover crops that were grown, and so this is using the CO2 burst method at various points of the year of those cover crop development. And you can see they're very different depending on the crop itself, but mostly across during the season it's very active. The CO2 respiration is different starting in November than it was July or early August. So it's going to change year to year too. If you get a rain event, if you till or something else happens, you add manure, that's going to change these things. And so again, you're always comparing apples to apples in these cases. So if you take a soil sample in your crop in early April, and then you take another sample down here in June, you might say, well, my health test, my soil health test was good in April, but then it was in June, soil health has decreased, and that's not the case, right? So some of these things, although they're measuring activity of healthy soil, not always those regions of what's working well. Okay, labile organic nitrogen was another test. This is measuring the organic forms of nitrogen, so what's coming out of that carbon again. These are the proteins and amino acids, those simpler forms of proteins that are likely to be mineralized as the microbes are active. The Woods End test, again, it uses a Solvita brand test called the labile amino nitrogen. So it's the same company that does the CO2 burst does the labile nitrogen, where Cornell uses a different test. It's the ACE protein index. And so when we look at those, again, compared to organic matter, and again, organic matter directly related to available nitrogen, Solvita test came out very tight, a curve of about 0.9. Cornell, the ACE test was about 0.85. So great correlation there at looking at simply organic matter, measuring that against nitrogen. Okay, so some of the other measurements they do
farmer-assisted research and management.
Probably most you're familiar with that, that's the ability of soil to resist erosion, to stay in those nice textures that we like plant roots to grow through. That
things that
it avoids crusting, slaking,
whatever
you know,
call it,
what
but
our soil clumped together. And this is very important for water infiltration, aeration, and rooting. And
stuff like that.
also very good for our microbes. This builds a good house for our microbes to live in when we have good water, air, and roots that are feeding them.
I'm gonna try.
So by doing this, you're also feeding or managing for microbes. This is a very simple test. This is looking at soils from conditions. These over here on the left are intensively managed. These over here on the right are more native soils. I believe this one was from a forest or a no-till system. And so these are dried. These are collected from the field. They're dried and they're crumbled. And then they're wetted. And then that dries off, and that's what you see at the bottom. That's what happens to the soil after it's wetted and dried. And so these with intense management, and we have these in our lab, and you can't even stick your finger in it. It's even hard to scratch.
It comes down
Of these are hard as rocks because these soils have no aggregate stability nor matter to act as a cushion to hold water, to push those back. And so those just dissolve and they form these very hard, almost like concrete compaction. Where over here on the right, these more native soils aren't disturbed, that don't have these methods maintained.
road is you.
And so again, directly related to organic matter. So when we look at versus Cornell, still a pretty good correlation, a little bit weaker
By the time
on Wood's End for aggregate stability compared to loss of ignition. It's about 0.58. And that's, again, it could be because of the loss of ignition isn't the most accurate measurement of organic matter. But with Cornell, when they looked at their test with that tighter organic matter estimation, their aggregate stability, they were able to estimate at about 0.9.
there was a lot of ideas.
Oh, and I just want to point this out again, just to say, you know, the tests are different where you have volumetric here and they're measuring that by weight there. So they're not always, they're just using different methods to look at the same thing. So available water capacity is another part of soil health that we're looking at. And this is the water that's retained between field saturation
And that's not what we're really interested in.
point that your plant is so depleted of water that it can't come back to life. And this is a function of pore size, and it's controlled by texture as well as organic matter. So texture would be something, the sand, silt, and clay level. That's an inherent property. That's what you've been given when you bought your farmland, right? So that's not something you can change. Your texture is going to remain your texture. However, organic matter is also a function of that, and it is a dynamic property that is something you can manage for. So this is a combination of sand, silt, and clay, as well as organic matter coming together. It's going to improve water. This is the equation for it. Basically, the take-home message here is for every 1% of organic matter, you gain about 0.17 inches of water in the plow layer. So you're really conserving your water there, and that equals about a half a day's worth of corn evaporation between during grain fill. You know, so basically just saying that your soil's more resilient, it's holding the rainfall that you're getting earlier in the season to later in the season when your soils are drying out and you're wishing it would rain again. So it's very good at holding the moisture, and we want to do that. This is probably one of the most important aspects we have in our cropping systems. Looking at soil chemistry, again, these are the well-standardized tests that we have available. These are for nutrients and pH levels, and these are extremely important for healthy crops. So they ran these tests basically, but again, they don't do it the same. The Cornell test uses a Morgan's extract. It's a very good test. It's well established. It's good for New York. It's good for northern New England. Even coming down to Pennsylvania, which is so far away, it doesn't test in the same way. You don't get as much accuracy as it does up north. Woods End uses the Haney 3, the Haney test. It's a fairly newer test. It hasn't been quite as well tested as the Mehlich 3, where the Mehlich 3 is the very standardized test that's been calibrated, correlated for quite a long time, and that's well published tests.
Like,
And so when we look at the nutrients here, the comparison between the Haney and the Mehlich 3 tests, for most nutrients it's really good. It's good for potassium, phosphorus, and magnesium. We saw a great correlation for all of these. However, when you look at calcium, calcium is not a good correlation, only 0.06% similarity there between those tests. Part of that is the Mehlich 3 test pulls calcium out of the soil, and so it doesn't give as good of a measurement as the Haney 3 does. So again, you're not, you just have to make sure your tests are comparing the same things, and that, there are differences between them. Okay, so after you go and you run all these tests, the labs will send you back a report. And it's a rating that you get.
with new clients.
Is healthy or unhealthy, and it breaks it down into different aspects. And so that's giving you the soil health indicators. How healthy is your soil based on this rating? And so we compared that, looking again at organic matter from each of the labs. A great correlation there but for Woods End at 0.9, and Cornell showing a 0.62 there. And these are the different things that are here. The reason that Woods End is so much tighter of a curve than Cornell test is because Woods End focuses mainly on those biological components and measuring those. Cornell looks a little bit more at the physical components, which, although are affected by organic matter, not necessarily measurable aspect of, or organic matter isn't something you would measure for these things. And so again, that's just showing just not quite of a tight of a curve, but you're still getting a 70.7% accuracy there, or comparability between those. Okay, so this is what an assessment looks like after you test, and this is on just the soil I grabbed, and so this breaks it down into all the different components. If it's green, it's good. If it's yellow, it's medium, and if it's red, that's where it's low, and that's where they make suggestions that you would change these. However, all this does is gives you an estimate of how your soil's doing.
Field scale, and so actually some of our field scale trials actually
Difficult to do. It's very general. And so after this test, you get this test on the first page, several tests later, and so I just grabbed this here. And so for active carbon, which is right here, it's about active carbon. It says why it's important, but it says your value 30 ppm. It corresponds to a score of about 65. This score is a medium range relative to soil, similar texture. Again, how does your test compare to other tests? That's all this is telling you. And it suggests it's not currently as strong, and maybe you boost up your management practices. If you had something like 100, we looked at one of those, keep doing what you're doing. If it's low, fix it. But again, just really, and the rating's different. I mean, here this rating says, well, your score is 65. This is from the Cornell test. And Woods End uses a completely different scale to measure their rating. So it's not as easy to compare, and it's just based on the lab itself what their rating system is. Okay, so there's some pros and cons of soil health testing. Based off of that, the pros are
no time frame as opposed to
processes. It's helping us push the envelope with our learning to get better at our research, to understand we know the microbes are there, what are they doing, how important are these? We know mineralization is important. We've been trying to figure this out for a long time. So they give us more tools, more options to look at these things. Overall, you can get a great report.
Cross factor, so there's more oil in soybean corn.
This might help you fix your situation. I'm wondering what this field's doing or what's going on here. So you can send this in and get a good estimate of what
still came out of it.
And it integrates different chemical, physical, and biological components. So really acknowledging that soil is a system and looking at how those things interact. So it gives us the ability to do that. Some of the cons are that the tests lack defined interpretations and calibrations that limited their use of management tool. For example, estimating the SLAN test doesn't give you a good recommendation for nitrogen application, whereas maybe the more standardized test does exactly that. How much nitrogen do I have to put on corn this year? The test packages are constantly changing, and I would like to add to that too, they're not similar to one another. They're expensive. I mean, is it worth $100 a soil sample in your test to get this? And how often do you want to do it? Maybe
Very tiny.
but maybe that's not something you want to do each year. And a lot of them have a long turnaround time. Cornell, for example, takes about 6 weeks to get those tests back because they're so intensive. So if you want to make a decision, if you collect your soil sample in spring and want to plant your corn in 2 weeks, you're probably not going to be able to do that based on these tests. And so I think
But
the soil test could be good or they could not work for you. But I also wanted to think a little bit about what we know about soil health and what we, maybe when we're thinking about soil health, evaluating it, assessing it, I mean, maybe it pays to get dirty. It pays to go out there and look. And I think we do this just in general. We tend to make things that an assessment of something, but forgetting to look at them. And I just wanted to come back to this. This is an old form that we have. It's Pennsylvania Soil Quality Assessment. And there's are available for every state. This just happens to be Pennsylvania's. And it does a comparison of just basically, this is the front page, and I know you can't read that very well. But this is the front page looking at all the different, you can characterize your fields, and then when you flip it over, what it does is it gives you all these evaluations, and it gives you, like it's a rubric. So you have different squares in there, and you read that. How do I assess my surface, my soil matter, erosion, compaction, infiltration, biodiversity, and plant growth? You look at all of these and you rate them, and you can go back year after year, and then you can compare them. So you're the ones that are out there doing the doing what's in the test, and it's probably a great thing to go once in a while and dig it up and take a look at what's going on. And so these basic papers, these basic rubrics are very important management tools too. And so some of the things you're looking at, does your soil have good structure? This is a great soil structure. You see the aggregates, large here, smaller ones clustered together there too. Again, lots of root growth. Are there organisms in here? Are you seeing earthworms? Are you seeing bugs and mites and things crawling? You can't see the microbes, but you can see the other ones. And so if you have a healthy soil, you're going to see them working around in there too. So just looking at it, this is a nice dark soil.
Got an idea.
Based on those, so depending on where you quite that dark, but this one's very high in organic matter. You can see it's holding a lot of moisture, lots of air infiltration in there. Compared to this is a soil crusting. This is a poor structured soil. You can see where he dug this out. It just plated up off of the top of the soil. It left a big hole there. There's not much structure here. In his hand, it looks rock hard. The soil, this little plant growing in here isn't very healthy, and it's not going to get good root growth through. And to look at that a little bit further, just doing a stability test. Sometimes we call this the Slake. And so this is a really, really simple procedure. You don't need anything fancy for it. You need a little bit of distilled water and a screen. And so in this sample, this is a soil, or they collected 2 soils and just put them in water. This one has good aggregate stability on the left. The one on the right, there's no soil in there, but there is later. Poor aggregate stability. So as that's dissolving in the water, you can see that with the good structure, it's breaking down. I think I lost, this is fading. This one's breaking down. It's forming in the bottom, but this one, look at how cloudy it's getting. So even though this is breaking down, it's breaking down slower, and as those aggregates are hitting the water, they're not dispersing. Where over here on the right-hand side, those are just becoming very cloudy and breaking down, and that's a really great test to do. So this might be a low tillage field, this might be a high tillage field that really destroys those structure. And so you can do this within a matter of minutes and look at your soil that way. Are there any signs of erosion or soil loss? This is a very severe field. You can see those rills. All the soil, look at how it's all piling up here at the bottom of this photo. It's very clumpy. You don't want your soil at the bottom of your hill. You want it on your hill. So looking at that, is there wind erosion? Is there water erosion? Involving in the crusting, the more
whether you do yield monitor,
you're going to have erosion. Are there signs? Again, a severe picture, but you want our soil to be springy. You want it to come back. Probably drive on it less often than you want to. and not when it's wet, and very deep rows or tire tracks here in this picture. And so soil penetrometers can measure compaction. You know, in most
I really thought it would be cool to do field
available,
mapping.
they're just pogo
I-
thing you push in your soil, and it tells you it hits the compaction layer, and this meter goes up to the red part of it, and that's where you hit your compaction layer. Typically, that's at the plow layer, as you can imagine.
Push power.
And break through that using different management practices to estimate compaction too, or actually measure it. Monitoring soil infiltration, how does your water run through the soil? These are some basic rings that you can pound in. Again, the NRCS personnel, extension people often have these. They can come out and help you.
The reason.
And it's measuring the quantity of water in a specific size ring, and you just put the water in, you let it filter through, and then you go and you do it again, and you measure the rate of infiltration. What you want to see there is you want to see that water go into the soil at a relatively quick rate. You don't want it to pond up at the top. So that's a fairly simple way of measuring infiltration that you can do out in the field. And look at your roots. You know, it's the plants telling you how healthy is your soil. You know, pull up a couple of them and look at your roots. This is a very root system.
Conventional.
Lateral roots, you have horizontal roots.
They're all
Tiny roots going in there, they're going deep. So this is a good example. You can see that holding on to those, kind of demonstrating what those roots are doing for the soil.
location.
The soil's doing for the roots there. And this is poor root growth. I'm sorry, this picture is a little.
Active response to that high.
This is soil that's been compacted. It has poor aggregate stability. So even though we're getting root growth through there, those roots are pushing between those two soil beds. You're not getting a lot of lateral roots. This is what we call conventional roots, where they're not coming out and they're not as branched.
We were looking at
Sure is.
growth space.
So all those little roots aren't growing as well. And so again, surface area. The more roots you have, the more nutrients you're going to get in your crops, the more water you're going to get, and the better it's going to help you build your soil. You can make your crops do your work for you while they're growing. And then here's just one more picture.
What was it?
Probably wouldn't be able to see this in the field, but I thought this was pretty neat. Over here on the left, very, very deep roots in a healthy soil coming down. Although these are still pretty deep here, not quite as deep, not quite as robust up at the top as these roots are here on the left. And then what else can you see? Are your crops healthy? This is not a healthy corn plant here. It's yellow, it's stunted. You can see looking around it, the soil's very compacted. It's starting to crack. That's a bad sign. So just going out and simple observations can be very helpful. So if you start managing for soil health, you might be surprised. I'm going to tell you to manage for organic matter. And there's different ways of doing it. These are the 14 spokes. This was a publication that was developed by Short-Diker at Penn State in combination with the NRCS. And so these are looking at continuous no-till systems. The most you can limit your tillage, the best. But there's also different activities.
You get down to a soybean.
You know, ideally you would do all 14 of these. Maybe you pick a set of them. Maybe all of these don't work for your farm, and that's okay. So a lot of them are very similar, but planting cover crops, diversifying your systems, rotating your crops, planting or using interseeding, all different things. Keeping living crops in your plants, reducing the loss of carbon can all help improve soil health. And this is that publication. This is
Have all the science.
if you want for it. And this is just a great little booklet for managing soil health. And okay, that's all I have. So I'm going to leave this up here in case you want to write that down. But I'd be happy to answer questions or take any comments that you might have.
And
You mentioned release of CO2 cover crops.
Mm-hmm.
CO2 is becoming most interesting than ever existed, apparently. How does the carbon sequestration that the cover crops accomplish counterweight that release?
Yeah. Okay. So the CO2 being released from the cover crops, how does that balance the carbon being sequestered balance that. So that was CO2 in the soil. That was not CO2 in the atmosphere. When CO2 is in the soil, that's going to be taken up by other microbes. They're going to use it. That's part of their energy system. And so they're going to keep it in there.
This should have
Soil, and so, I'm sorry, to answer your question fully, you're getting all
economics,
above ground. That's the question
the extra.
there. Carbon is very stable. It's tied with other carbon molecules, which is a very strong form. That's the organic form of it. Has the nitrogen bonds in there, and so that's the sequestration. So then build that into your soil when that gets plowed in, and that's going to be stable. As the microbes eat that, they're going to hold it in their body, sequestered.
26,000.
As you disturb that soil and you release that CO2, it goes into the atmosphere. That's where it becomes damaging. But when it's in the soil, it's fine. Yeah.
Bill Rohr, we've run hundreds of samples through Aggregate for soil health. One of the greatest challenges that I see with soil health is a direct relationship with low organic matter. And for most of the samples that we've run through the lab, we're looking at 1% to 2% organic matter. And the rating's always been very low, regardless of the practices that are being implemented on that ground. So we have some of the best ground on the Delmarva Peninsula with cover crops, a little bit of manure here and there, with all kinds of no-till practices, and they still come up with very, very low ratings. So that's my greatest challenge with soil health evaluation.
Great.
Is it really puts sandy soil to significant disadvantage to how we look at soil health across America.
Great. I'm going to repeat that in case people in the back couldn't hear it. He works in a laboratory, and he works with sandy soils in Delmarva, so they're just naturally low in organic matter. When he runs his tests, the soil health test, he gets very low estimation, like these soils aren't healthy despite the practices. They're doing all the right things, they're managing their carbon, but when they get their soil health test back, because of those inherent properties of the soil, it's high sand, low organic matter, it's going to come back with a little bit low estimation of the soil health test. Is that a good summary?
My point is, at a national strategy that you're involved in, really, there needs to be a calibration in place.
Yeah. Yeah, so across the nation, there has to be a better calibration, and that is a problem. And I have friends out in Washington who are saying, "Oh, well, we run the soil health test, the Cornell test on our soils out here, and it's not accurate either." So it is. That's a really great point. That it is kind of place-based, and it works.
We try to determine.
It isn't universal, and so that's an area that we need to explore a little bit more as scientists and being able to estimate some of those things. Thanks.
Are you comfortable with any bioassay tests, when you're playing out actual microorganisms and how that relates?
Sure. The question is, am I comfortable with bioassay tests, microorganisms, and how that works? I don't do that specifically. My thought on it is very complex. They're very highly technical. So yes, they're a great way of looking at the microorganisms, but they're still developed too, and there's a lot of variability among those, and the scientists are trying to get better at assessing those. But again, you have microbes, and they're extremely complex organisms in themselves, so to break them up and to look at them, it's a very big challenge. And then once you get up, what are you going to say? So some of the tests are good. They can look at, well, here's a population of bacteria, versus, and then we have this population of fungi and something else. So they can tell you there.
Sit there and we can plant sample.
Different bacteria within that, and so they're better. They're doing sequencing on that. So that's evolving, but right now it's highly technical. Some of the folks that are doing it at Penn State, you got to, they can make to kind of take in what they're talking about, because it's very technical in the lab based research. But it's evolving. It's evolving, and I think.
Excuse me.
The strong correlation between organic matter and these other tests. What's the motivation for doing that?
Yeah. So because of the strong correlation between organic matter and the other tests, what's the motivation for doing the other tests? The other tests help you see the whole, your soil as a whole. To see is it respiring? Am I getting the nitrogen? Am I getting aggregate stability? Is that worth it to you? $100 worth it to you to understand that? I think that's something you have to determine place-by-place basis, whether it is or not. I don't know the motivation just to understand your system as a whole and to start looking at certain things that aren't there, or that you might not get in some other.
Soybean rotation.
But there is a strong correlation between these matters. So if you want to know a lot of these things and you would rather spend $12 than $100,
Or
it's a better motivation.
water. Actually.
Well, thank you for your time today. Thank you.